Hello everybody, my name is David Moyano-Palazuelo and today I'm going to share with you our work called T-Rex, a Tandem Repeat Explorer algorithm for the fast, accurate and simple detection of tandem repeat protein based on their secondary structure. So, let's go. Tandem repeat proteins, or TRP, are proteins characterized by sequential repeating units known as tandem repeat units or TRUs. Those TRUs can be superimposed in the three-dimensional space. TRP are found in a broad range of organisms and are very diverse. Significant examples of TRP include the beta-propyl folds, which play a crucial role in protein-protein interaction and various cellular functions, and armadillo repeat, also involved in mediating protein-protein interaction. Despite their biological relevance, identifying TRP is a significant challenge due to the high sequence variability within TRUs. This complicates traditional sequence-based detection methods. Additionally, while tertiary structure-based methods can provide valuable insights, they are typically time-consuming and not ideal for large-scale data analysis. In response to this challenge, we present a novel, efficient method for identifying TRP based on secondary structure. As you can see in this example of P49951, the secondary structure is conserved between the TRUs, in contrast to the amino acid sequence. This property allows us to detect them using the secondary structure. The T-REST algorithm has two main steps. In the first step, the algorithm starts identifying the repeat pattern in the secondary structure. To do that, we first calculate all the possible substrings inside the secondary structure sequence. Then, with all these substrings, we evaluate them, multiplying the absolute frequency of each substring by the length of each one. After that, we selected the substring with the highest score. With the highest score substring, we scan all the secondary structure sequence, identifying regions of high similarity. The coordinates of the region with a value higher than the threshold are selected like the coordinate of the hypothetical TRUs. This last step lets us introduce some variability in the TRUs in the process. After the first step, the previously identified regions are evaluated three-dimensionally. To achieve this, we first structurally align this region. Then, we calculate the values of the TM score and the RMCD, obtaining a matrix with the TM score values. Subsequently, the values of the matrix are filtered out, eliminating those coordinator regions that, on average, do not exceed the cutoff values. Thus, we finally obtain the coordinate of the TRUs. Next, we compare the performance of the algorithm with a reference dataset, in our case, the training dataset of another popular method for detecting TRP, Raphael. Specifically, the high specificity of our algorithm stands out. Almost all the TRP detected as such are indeed TRP. This high specificity has a cost, which is the low sensitivity that the algorithm currently has, being only able to recover a third of the protein with TRP in the dataset. The algorithm is useful for detecting TRP, also identifying the coordinate of the TRU within the TRPs. Here we saw two significant examples where the algorithm works very well. In the first example, we see a tetratricopeptide repeat, while in the second, we see a leucine-rich repeat. These are just a few examples, but the method has been able to identify many more repeat proteins of different types. Specifically, the algorithm was able to find 456 TRP in 10,000 proteins from Drosophila in just around 3 minutes. To put the extraordinary speed of the method in context, we can compare it to the RepeatDB Lite, one of the best performing methods. This one takes a range of minutes to analyze just a single protein, depending on the protein. With this performance, it can be used like a super fast pre-filter. In conclusion, our algorithm has a high specificity but low sensitivity. It is useful for detecting new TRPs and it showed very promising preliminary result in predicting the coordinate of the TRUs on a larger scale, both in performance and in time. As for the future, we hope that we can soon deploy our algorithm across AlphaFold database. That's it, in a database of over 50 million proteins, allowing a large-scale study of the distribution of this protein in the tree of life. Improvements are needed for the detection of various repeated patterns within the same protein, as well as strengthening and fine-tuning the algorithm to enhance its sensitivity, which require a better understanding of the intrinsic variability of the TRUs. And this is all. Thank you very much for your attention and see you during the Congress.